First up, we have Luca.
Thank you. Thank you, Philip, and thank you, everybody. Good afternoon. Now we are supposed to start with, where can I find my...
Just close that screen.
Yep, here we are. Oops. Very sensitive. Very sensitive. Here. Okay. Yep, perfect. Thank you. So I would like to embark you today, ideally, on a journey to Antarctica, more specifically to Victoria Land, which is in the continental Antarctica, and that belongs to that 0.35% of Antarctic surface not permanently covered by snow and ice. In Victoria Land, soils are characterized by permafrost, generally located at a depth between 20 centimeters and 1 meter. And generally speaking, they are supposed to be characterized by very simple food webs. And this is essentially due to two main reasons. The first one is the harsh climate. The mean annual air temperature is between minus 20 and minus 14. And the mean summer air temperature is between minus 6 and minus 2. In addition to that, rainfall comes only like snow, equivalent to 100, 200 millimeter per year. The second reason is that the major autotroph plants, just to be clear, are here represented only by cryptogams, mosses, and lichens. We don't have vascular plants and what this means in terms of rhizosphere processes and so on. In parallel to this, in the recent decades, there has been an increase of the thickness of the active layer in this permafrost soil, which has been also accompanied by a change in the cover of mosses and lichens. So on this scientific background in mind, the idea of this study was essentially trying to answer one major question: how does a change in cryptogam abundance, first of all, drive the amount of soil organic matter that we can find in these Victoria Land soils, and how this feeds back on the microbial community. Trying to answer this question, first of all, we sample a certain number of soils in 9 different locations in Victoria Land, trying to follow a latitudinal gradient, so to take into account a geographic position, about 4 degrees of latitude, and also under different bedrocks, including igneous, metamorphic, and sedimentary rocks. The data that I will present are still under process. Actually, they are preliminary in the sense they are very fresh, not only cold, very fresh. And they refer, of course, to the first 2 centimeters of the soil depth, where most of the organic matter, of course, is accumulated, if it accumulates. And in the field, of course, there are some permanent plots where soil samples have been sampled, and where, of course, estimation of lichen and mosses have been carried out. Once soil samples went back after a long travel by ship and planes, they went back to the laboratory. Of course, classical chemical analysis in order to determine the amount, first of all, of soil organic carbon, water content, major nutrients, pH, the microbial biomass nutrients in terms of carbon, nitrogen, and phosphorus. And here, to have an idea of the microbial community structure, we use the phospholipid fatty acid analysis, thanks particularly to Bjorn. And then we also measure the extracellular potential enzymatic activity of about 9 hydrolytic enzymes and 1 oxidative enzymes. So let's start to see some of these fresh data. And this is a correlation circle, thanks to Vincent here for the statistical analysis, that try to correlate most of the variables that we have been able to, to measure in the field and in the lab. And there are 3 points that I would like to highlight here. The first one, as you can see, with increasing of moss cover, we have an increase of soil temperature, water content, and a set of nutrients. Keep in mind these two parameters, soil and water content, as well as the increase of nitrate, ammonium, and phosphate. So it means essentially that mosses are the main drivers, of course, to enhance this accumulation of soil organic matter. The second point that I would like to highlight is the fact that in some way, once organic matter starts to accumulate, this superficial soil area is independent from, for example, the local geological condition, the lithology, and in some way also from the microclimatic conditions. So they are in some way detached from the local geographical and geological characteristic. And the third aspect is related to the pH, which is actually significantly correlated to the accumulation of organic matter, but in a negative way. It means that with increasing soil organic matter, we have a decrease of the pH. This graph also gives you an idea of the wide range, actually, of variation of the pH along this cryptogram gradient of biomass cryptogram gradient. So what happens in terms of microbial community structure? So here you have the ratio of the fungi and the bacterial biomass based on the phospholipid fatty acid, and essentially this is the 1-to-1 ratio. Essentially, this tells you that in most of the soil samples, actually bacteria are the dominant microbes in these soils, but fungi start to be relatively more abundant with increasing accumulation of the soil organic matter. And this is actually in line with a recent paper that has been a recent study that has been carried out in maritime Antarctica, showing the distribution and the diversity abundance of fungi, and showing that temperature and water availability was the main drivers. And actually, if you remember before, here, actually when we have more soil organic matter, we have also more water content and also a higher soil temperature. So, and this will promote not only the survival of fungi, but potentially also spreading their development. Increasing soil organic matter, of course, causes also an increase of the microbial biomass, carbon, phosphorus, and nitrogen. We have ideally, potentially, a higher microbial community. And necessarily, to face this increased necessity of nutrient, we have in parallel, overall, an increased total enzyme activity for enzymes targeting carbon, nitrogen, phosphorus, with increasing microbial biomass, carbon, nitrogen, phosphorus, particularly for carbon and phosphorus targeting enzymes. So at this point, we decided to make a kind of, to play a little bit with the data and to calculate the ratio between the total enzyme activity on the corresponding microbial biomass nutrient, what we can, for example, call mass-specific enzyme activity, and relate this activity, enzymatic activity, to the major environmental parameters that we showed before. And what came out is that in the case of nitrogen and carbon, this is particularly beta-glucosidase. This is the sum of all the 5 nitrogen-targeting enzymes. You see that in the case of the carbon, the beta-glucosidase, there's an inverse relationship with the pH. So when the pH is more acidic and we have more organic matter, of course, this carbon-targeting enzyme is also higher. An opposite trend we have for nitrogen, where indeed the activity of the nitrogen-targeting enzymes is higher with a higher pH and a lower organic matter. In some way, this is not new, but this is nice because on a smaller scale, what we are observing here along such a huge, relatively huge pH gradient is what has been observed on global scale, looking at the activity of the enzymes, and actually confirming that beta-glucosidase is more active when we have more organic matter and lower pH compared to the nitrogen-targeting enzymes. Now, we can debate what's the main reason, if simply an effect of the pH, or there's potentially an effect also of the quality of the organic matter. Here we have cellulose. Here we don't have cellulose. We don't have plants. We don't have mosses, excuse me. So could be the activity in this case of the nitrogen targeting enzymes provide not only nitrogen but also carbon, of course, for the, for the microbe. So it could be interesting to give probably a deeper look on the quality of the organic matter that we may have in these two different extreme situations. For what concerns the phosphatase, the phosphorus degrading enzymes, the best explanator is an abiotic variable, the ratio between the fungi and the bacteria biomass. Increasing the relative amount of fungi, we have also a higher activity of phosphorus, the phosphatase. And this, again, in some way confirms the potential higher capacity of fungi to solubilize phosphorus compared to bacteria. For what concerns the oxidative enzymes that I mentioned before, again, this is not new, but it's still nice because it confirms what has been, again, observed on global scale, including different type of ecosystems, the direct relationship between the phenol oxidase and the soil pH, of course, being higher with more basic soil. So, in conclusion, what we have obtained until now is we have seen until now is a primary role of mosses in controlling and driving the accumulation of organic matter in the Victoria land soil. Higher content of organic matter, of course, is associated with an increase of microbial biomass nutrients, and of course, this will boost a higher activity of the enzymes. At the same time, bacterial biomass is dominating the microbial biomass. Fungi, they start to play a major role with low pH and increasing organic matter content. Mass-specific enzyme activity is differently controlled in the case of carbon, nitrogen, phosphorus-degrading enzymes. And again, the oxidative enzyme is, again, controlled by the pH. Let me finish here just thanking the Italian research program in Antarctica for funding this research. And I would like to know if there's any Norwegian colleague today here. No? Well, we should congratulate with a Norwegian colleague because exactly 105 years ago, actually Amundsen and 4 other Norwegian explorers were the first to reach the South Pole. It was the 14th December, 1911. Unfortunately for you guys, 5 weeks before the British explorer, Robert Falcon Scott. Thank you very much.
Thanks so much for that, Luca. Fascinating. So it's open now for questions. I think we've got time for 1 or 2. Okay, what do you think? Okay, right, excellent.
Microbial biomass data, really quite high, surprisingly to me, how high the microbial biomass C was.
In terms of, you mean nutrient or in terms
No, in the biomass of carbon, like tens of milligrams per gram, a lot higher than I thought. Is that
on the side of more, let's say on the more cryptogram side?
Yeah, well, just generally, like across the data was rather high.
Well,
and I just thought, is that it's quite interesting. Sort of confirms this idea that most soil organic matter is necromass, basically.
Okay, let's say that thinking to the samples actually on the. To be clear, on the moss abundant side, actually the first 2 centimeters, it's actually primarily moss biomass. So that's the reason why it's very high. But on the other side, actually, I would say it's relatively high in the sense that it's on the range that has been observed also for other soils in Antarctica. And while we can debate if it's high, personally, I don't think it's super, super high. But of course, we have still some let's say, not only microbes, but there's still some cyanobacteria that can partly explain the values. There's maybe the transport by wind of organic matter from other parts, I don't know, the bones or wheels and so on. But again, it could be interesting to see, besides the abundance, also the quality, how it differs along this gradient, how can explain the differences.
I think we probably should, if you want
Thank you.
to ask Luca any questions later, please capture it. So we should straight on now to Sigrid, and I'll see if I can help you with this. Oh. Okay, Sigrid.
Good afternoon. I will present to you part of my work on mycorrhizal networks in plant community dynamics. So, when former agricultural land lies fallow, pioneering plant species will start to cover the soil. And when time goes, then also the plant community will start or continue to develop. And new plant species come and other plant species go. And this dynamic of plant community establishment, it can be partially explained by the microbial community in soil. When plants grow, they shed litter onto the soil and their roots excrete root exudates, and this accumulates a unique microbial community around the roots, which is specific for plant identity and the age of the plant. But when this plant dies, then it leaves a legacy into the soil. This has an impact on the next plant that will occupy its place. And this is what we call plant-soil feedback. That can either be direct when it has an impact on the same plant species, or indirect when it does impact another plant species, and it ranges from negative to positive. It can be strong, it can be weak. And the majority of the plant species have a negative plant-soil feedback, which also include crop species. For example, one cannot grow potatoes on the same field year after year because of the accumulation of pathogens. But also during succession, we believe that plant-soil feedbacks play an important role in the community composition of the plants. It is known that over the whole range of the succession, the soil microbial community does change. And in this example, we can see that in this one, when you have early successional species, at first arrival they perform very well, but the next generation does not perform so well. This is a negative plant-soil feedback. But when we look at the late successional species, we see that they need one generation to accumulate a beneficial environment. And that is a positive plant-soil feedback. So we see that both the plant identity matters, but also the soil microbial community matters. So you see here that soil origin does matter as well for plant performance. And one possible explanation here is the action of the mycorrhizal in soil, which are plant symbionts. So these mycorrhizae, they can form a network in soil connecting several plant species. And the plant provides the fungus with carbon, and the fungus provides the plant with additional water and nutrients. But these resources are not always shared equally among the plant species. And in that way, it can also have an influence on plant community composition. And this led to my research question, is can we find a role of these mycorrhizae networks during succession? So these are my hypotheses. Because AMF is a symbiont, I expected that if I would interrupt this mycorrhizal network, that plant biomass would reduce. And secondly, we've seen that the soil microbial community does change during succession, and therefore I hypothesized that the AMF networks in early successional stages contribute less to plant growth than in mid-successional stages. I've also shown that plant identity matters, and therefore I hypothesized that plants with positive plant-soil feedback are more negatively affected by AMF network interruption than plants with a negative plant-soil feedback. I did test this with the core rotation experiment in the Netherlands. On the Veluwe area, this is in the middle of the Netherlands. I had 3 plots with early successional stages, those orange ones, and 3 with mid-successional stage plots. And I had 8 plant species which I tested, 4 grasses and 4 forbs. And I also split them up according to the plant-soil feedback. So 4 of them have a positive plant-soil feedback and 4 of them a negative. And the negative plant-soil feedback means that those species, they prefer to grow in soil conditioned by other plant species, or those with a positive or neutral plant-soil feedback prefer to grow in soil which is conditioned by themselves. The plants will grow in metal mesh tubes that you can see here. The plant roots cannot go through, but mycorrhiza and other microbes can cross. Inside the tubes was plot-specific soil, and in order to interrupt the mycorrhizal networks, I rotated the cores every third day. The experiment took just a bit over 2 months, and after the experiment I did harvest the below ground and above ground biomass. So the first results on the x-axis, you see the 2 plant functional groups, and this is above ground plant biomass, and we see that the treatment, so the rotation, did not have an effect neither for forbs nor grasses, for the negative plant-soil feedback plants. However, for the neutral to positive plant-soil feedback plants, we do see an effect of the core rotation. And not for forbs, but for grasses. And we see that when those tubes were not disturbed, so not rotated, they had lower biomass than when I did rotate the tubes. We did not find an interaction between the treatment, so the turning of the tubes, and successional states, but successional states did matter for biomass production, not for forbs, but for grasses. You see that they perform better in mid-successional state soil. So to sum up, so the interruption of the AMF network did not reduce plant growth, neither did I find support for the difference in successional states on the mycorrhizal network effect, but we did find support for the third hypothesis. Thus the plants with a positive plant-soil feedback were more affected by the treatment than plants with a negative plant-soil feedback. So the result was a bit in contrast to what we expected. However, other researchers also found an increase upon disturbance of this AMF network. That was either because those were seedlings, or these plants are not strong sinks or not good traders of carbon. We also only found an effect for grasses and not for forbs. This might be due to the nutritional needs. So forbs are stronger phosphorus sinks, while grasses are stronger nitrogen sinks. It might be that the soils were more nitrogen limited and phosphorus limited. But additional work on the soil analysis will show this. And next thing is that we only found effect for those plants with a positive plant-soil feedback, and we know that plants with positive plant-soil feedback have higher percentage of mycorrhizal colonization. And I will have a look at this as well, and maybe we can also find a difference in mycorrhizal colonization between the 2 functional groups. And then the last thing is, it will be nice to know where this carbon actually does go to. So those plants that have reduced biomass, do they produce carbon at all? And is this extra carbon transported by the AMF network either to other plants? And does this change when these plants become bigger? Do they become proper carbon sinks then? So to take home, the effect of the mycorrhizal network is interruption depends on plant-soil feedback and plant functional group. I'd like to thank all the people that helped me out with the setup of the experiment, the maintenance, and the analysis. And thank you.
Thanks for a really neat presentation, Sigrid. That was great, keeping lots of time. Any questions? Okay, so while folks think about that, I just wondered whether or not, if you changed the nutrient status of the soils that you were starting with in the experiments, that might have perhaps interacted with the results. I guess the site history is probably really important here, isn't it?
Yeah, they're all former agricultural lands. And I'm still working on the project, so I hope soon I have the results of the nutritional status of the soil. And I agree that that will definitely have an impact. But I don't know yet in its direction. I can imagine.
I guess I'm wondering whether actually if you were dealing with unimproved meadow systems that have not had a history of sort of fertiliser application, whether you'd see stronger linkages here and mycorrhizal effects.
Again?
I guess if it was unimproved systems, so that haven't had maybe years of fertiliser inputs, whether you perhaps see a stronger relationship between mycorrhizal status and plant performance.
That might be, yeah. But I would also expect that with later successional stages, you have these interactions become stronger anyway.
Yeah, absolutely. Yeah, we've got time for a couple of questions, yeah.
In your experimental design, when you're doing the rotation, do you think you're interrupting only the avascular mycorrhizal fungal mycelium, or would you interrupt also other fungal mycelium?
You interrupt all fungal mycelia that grow through, indeed. So it might be that I'm not only looking at the effect of arbuscular mycorrhizal networks, but also maybe pathogenic.
Exactly. So could it be that your results are more explained by you interrupting the non-beneficial fungi in the soil?
Yeah, that can also well be. Yeah, but I think, or at least I would expect that it has the biggest influence on mycorrhizal networks, because if you turn them, then you at least break any connection between this plant and maybe other plant species, while other fungi might not be directly connected to other plant species. But I agree that there will be other fungi affected as well. Yeah.
Time for one quick one. Yes, Tom.
So with the rotation that you're doing in the field, do you think there's a risk of disturbing the soil outside of the cores and then therefore stimulating some kind of release of nutrients there? Your results?
There has been some research done on that, what the effect is of these tubes in the soil, and they found it has no effect on nutrients in the soil. But what I have seen in a parallel experiment where it was really dry, then if you disturb it, then you create like an air layer between the tube and the soil. And because it's so dry, it does not fill up again. So I think that's one of the disadvantages. But in this study, we had perfect weather actually, so it was not really dry, the soil. So I did check whether the soil connected well to the tubes, and that was fine. Yeah.
Okay, we probably should get ready for the next one. Thanks so much again. That's good. That was really good. So next up is Maria. So whenever you're ready, and I'll see if I can find your presentation. That's always good. Here we go. Now then. So where am I in terms of? Oh, there we go. Yeah, different format. It's all different. Okay, thanks.
Good afternoon, and thank you for being in this session. I know there is a heavy competition for this time. So I'm trying to tell you a little bit about the species pools and the dark diversity of avascular mycorrhizal fungi in the next 10 minutes or so. What are species pools? Here I'm showing original schematic summary of the species pool concept as it was proposed by Martin Sobel some time ago now, 1997, in Trends in Ecology and Evolution. And what he was positing with this concept is that the actual species pool, meaning the local observed species diversity, it's determined by many processes acting at other spatial scales than local, meaning the regional spatial scales. And actually it begins with speciation. So any species locally present can be present if they have evolved and if they have evolved for these conditions. And then of course they need to migrate and disperse, large scale and small scale dispersal. And at any locality they are going to face filtering, abiotic filtering and biotic filtering, so they need to survive the conditions that they are going to experience. So that's their theory. That's just to summarize this theory. So in brief, the species pool concept is saying that the local variation in diversity of organisms is going to be determined more by regional scale factors. What do we know? Sorry, before we go into what do we know about this, I'm trying to bring it a bit closer to you by an example, which is maybe easy to understand for people used to work with plants. But let's imagine two habitats, a raised bog and a grassland, quite common habitats over here in UK, but also in many other temperate regions. And we do know that in these two habitats, we have quite a different plant diversity. So grasslands, we're used to know that these are very diverse. There's many plant species that can grow there. In raised bogs, quite the opposite. So the observed diversity in two types of habitats is very different. It's small in one and large in another. Does it actually matter? How many species can grow in these habitats? So we do know actually that the number of species that have evolved to stand the conditions of raised bog is quite small. So the species pool of the raised bog is quite small. And the species pool of the grasslands, quite the opposite, is quite large. So in a way, when we compare these numbers, small number of species or large number of species, it's really not relevant. But from there we can go on and we can ask, are all the species of the species pool of the raised bog present in this locality of the raised bog? And if they are not, then they belong to what we call the dark diversity. So it's the missing members of the species pool that could grow in the location, but we are not observing them at the moment in whatever reasons. And so we could observe that the small, dark diversity could be small or large in any of these habitats, and this way also the completeness of the communities, meaning what proportion of the species that can grow in this habitat we actually observe in the locality at the moment. So the completeness also can be small or large. Why it would matter to separate the diversity into such components? And that's because these components of the diversity may respond to different factors. They might be determined by different mechanisms. So I say that there are three components of the diversity. One of them is observed diversity, the most simple one. Another one is the one we don't observe, the ones that are missing. And the missing ones and the observed ones are making up the species pool. So, with this knowledge of the terminology, I'm going to go into AMF world. And I'm first asking, do AMF have species pools? Why is this question meaningful? If all AMF could grow in all habitats, then they don't. Then we have one global species pool. And actually, it would make no sense for me to ask, do the species pools differ in any place? So let's see what we know about that. Here at the left side for you, left panel, we have some global data on AMF diversity. And this is plotted, richness per site is plotted against pH gradient. And it's separated for the temperate and tropical habitats. And we see here that the richness, AMF richness per site, responds very differently to pH gradient in the tropical and temperate locations. What is it telling me? It's telling me that tropical and temperate AMF have evolved differently in regards to their habitats. So here's first indication that AMF do select the habitats, they do differ in terms of where they can grow. In the recent years, there's been a big increase in knowledge about global patterns of diversity of AMF, and one of the pieces of knowledge comes from our team, which was published in Science last year. This slide summarizes what we learned. So we collected samples globally, sequenced AMF in plant roots, And this slide is showing how many species, I'm using here the term virtual taxon or VT, which is a molecular species, how many of them occur in any continent and how many are shared among the continents. So if you see the numbers, or maybe you don't, then the continental diversities are quite similar. But also the proportion of AMF what's shared among the continents is quite similar. It's about 50% in many cases. But the most interesting thing we found there was that many AMF are present on multiple continents. At the same time, it does not mean that everything is everywhere. We had regional differences. So when we go deeper into the regional patterns, then we see that actually there is a latitudinal diversity gradient for AMF local richness. And curiously, we saw that this latitudinal gradient differs for major biomes. It differs for grasslands and forests. So the dark symbols here, the black ones, these are grasslands. So grasslands all over the globe seem to be more diverse in terms of AMF than the forests. At the same time, both show the latitudinal richness gradient. Tropical areas seem to be more diverse. Now when we use this global data and start mapping the local richness at the global scale, we can actually also compute the local diversity, species pools, and the dark diversity. So I'm trying to walk you through this. I can say that behind this richness map actually is a lot of local scale richness data, and we did use the Anchau's way to standardize the data. So it's the extrapolated, rarefied, etc., etc. So we took great care to make the richness data comparable. So when we plot the local diversity, you see that some parts of the world are more rich. So Africa, South America seem to be more rich. Okay. What about the species pools? So we can compute the species pools, and I'm not going to go into the algorithms here, which are quite complex, but Meelis Pärtel has developed a lot of this. So we see that the species pools show quite similar pattern, but not exactly. So the species pools again higher in some tropical areas, but not exactly in the same places as the richest locations in terms of local diversity. This, of course, means that the patterns of the dark diversity, this missing part of the species pools, is different. That's nice, but what is generating this? And what is generating this, I'm not telling you yet, but please stay tuned to see the paper in progress, because that would be most interesting. Map is also nice, of course. Let's go a bit further what we know and how we understand the determinants of the local diversity. And here we used the same data of our science paper, and we put it through some complex statistical analysis, randomizing, computing phylogenetic diversity, etc. And we could use this to test which factors are influencing AMF at these different spatial scales in relation to the species pool concept. And I'm summarizing the very complex tests in the simple graph, and we could find evidence for the dispersal limitation, dispersal properties of AMF operating at the higher, larger spatial scales. So it does matter what are the properties of AMF in terms of dispersal. What the properties really are, we need to find out further. We found evidence for habitat filtering operating, and there was also evidence at the more local scale that the host is a filter that is influencing which AMF are present in any location. So I'm jumping a bit more deeply into the dispersal in the last few minutes I have. Dispersal of AMF is something that we need to understand a lot more, but there is quite a little empirical evidence of them. So briefly, they have three types of propagules, three types of vectors. Which of these propagules are more important? Which of these vectors are more important? We really still don't know. But I'm showing you two case studies from our team we have made recently. So the first one is looking at how AMF can arrive to a new habitat patch. So here is a landscape within a grassland, natural grasslands, some forest patches, and the white parts are limestone quarries. So we placed into these limestone quarries boxes like this. So it's a new habitat, and we sampled then. That was sterilized soil. There was no AMF in there, and we sampled these boxes half a year and 1 year later, and we could identify in these boxes actually quite a number of AMF species, 30 species in total. And curiously, most of these species were, large part of these species were ruderals. In terms of abundance of sequences, most of these sequences were ruderal AMF. So these ones that can easily disperse. Curiously, the number of plant species that arrived in the same time was the same. So in a way, I can say AMF do disperse as well as plants. In the same study system, we have also followed AMF in the secondary succession. And what to take from this little bit complex slide is community completeness of AMF, which are these purple stuff, in young, intermediate, and mature successional stage of the vegetation. And the plants are green. So community completeness, the higher it is, the more of the locally, regionally available species pool have arrived to these communities. And look at the young stage. So AMF community completeness is higher than that of plants. At the intermediate, AMF community completeness is higher than that of plants. At the mature stages, they are the same. So in a way, in the secondary succession, AMF actually kind of arrive more efficiently than plants from the regional pool. With this, I'm done. So I hope I made it a bit clearer what are species pools. I say that AMF species pools are larger in some tropical areas. Stay tuned for the reasons for this. AMF do have global diversity structure. Not everything is everywhere. Dispersal limitation is clearly present, and of course we need to understand way better how they actually disperse. Thanks for the team, for the financiers, and thanks for you for listening.
Thanks very much for that, Claudia. So over to the floor. Any quick questions? Please.
What happened to the species pool of North America? There was a guy kept there.
Yes, it's quite curious. No, I can't really explain why is the thing in America looking so much less diverse. Yeah, I don't have a good explanation for that. Was there another?
Is there some limitation of for the global scale, and that a lot of places just haven't been sampled efficiently, clearly enough?
Yeah, I agree. I mean, part of this understanding, of course, could be due to incomplete sample size, incomplete coverage of the observation points. But I can say that we actually quite thoroughly have looked into this, whether when we remove some sample sites, then how much the pattern changes, and it's not a big influence. So in this way, what we have been finding about the species pool patterns, it's not really due to incomplete data that much, but it quite well reflects what's probably biologically true. Yeah.
Okay, thanks so much. We probably should press on at this stage, so we'll do talk to Maria afterwards. Thanks very much for that. So next up we have Cesar.
And there we go. That's right. Yeah. So view, and then full screen mode.
Thank you. I'm going to present you part of my PhD thesis. So I'm working in southern Chile. Located right here. These temperate forests correspond about 50% of the temperate forests of the southern hemisphere. But they have big differences with northern hemisphere. For example, there is extremely low contamination, and there is a lot of endemisms. Also, the last glacial maximum occurred in this limit and covered most of the Andes in the region. So the soils in the Andes are really young and really volcanic soils, richer in nutrients, while the soils in this system, in this mountain system, in the coast, are poorer, but there is a strong oceanic influence in this forest. Also, we have two major forest types: conifer forests, native conifers, around 10 species, and nothofagus forests, that they are 7 species. These conifer forests are associated with arbuscular mycorrhizal fungi, while the nothofagus forests are associated with ectomycorrhiza. And the cover in these forests is around 80% in the conifers and 90% in the nothofagus forest. So they are the dominant element. Furthermore, just from the vascular species plants described there, just 7 species are associated with ectomycorrhiza, and these are the 7 species corresponding to the nothofagus genus. So we ask 3 questions. We ask how are the difference between fungal communities in ectomycorrhiza and arbuscular mycorrhiza dominated forests, I mean nothofagus and conifer forests. We ask if these fungal communities differ in Andes and coast, and we ask how are given these differences by different fungal guilds, not just the fungi community as a whole, but different fungal guilds. So to do this, we select 5 sites and 13 plots overall. In each one of these sites that correspond to national parks, we compare nothofagus forest versus conifer forest, and we had in overall 13 plots. We sequence soil, bulk soil, with Illumina and ITS2, basically, and these are the main results that we had at the functional level. So we can assign each one of these OTUs at fungal guilds. For example, in the green, you see here arbuscular mycorrhiza, then brown root, then the big yellow bars are ectomycorrhiza, and the big blue bars are saprotrophic fungi. So the main result here is that in arbuscular mycorrhiza dominated forests or plots, saprotrophic fungi is highly abundant, and ectomycorrhiza is present but is not that abundant. And of course, in ectomycorrhiza dominated forests, ectomycorrhiza is highly abundant. But if we look at this at the proportional abundance or the proportional richness, then we can plot these 2 abundances of ectomycorrhizal fungi and saprotrophic fungi, and there is quite a strong negative relationship. And this relationship is given more by the forest type that is in the empty symbols than in the mountain system. So if you saw here, the arbuscular mycorrhiza plots had a higher proportion of saprotrophic fungi and less proportion of ectomycorrhizal fungi. Then we can compare the different community structures of the different fungal guilds, first all fungi, and then saprotrophic, ectomycorrhiza, and arbuscular mycorrhiza. We can run a permanova to see how these factors, basically the mountain system, Andes or coast, or the forest type, ectomycorrhiza or arbuscular mycorrhiza, structure these different communities. And for all fungi, saprotrophic and ectomycorrhiza, both the mountain system and the forest type are important in structuring the communities, but in arbuscular mycorrhiza, just the mountain system structure these communities. Then we can do the same kind of analysis, a canonical correspondence analysis, to see how the different edaphic factors affect these communities' structures. This is a summary of the 4 canonical correspondence analysis. And the main result here is that arbuscular mycorrhiza, saprotrophic fungi, and all fungi in general have more or less the same edaphic factors affecting these communities, while ectomycorrhiza is just affected by pH and potassium. So you see, for example, arbuscular mycorrhiza, the result is interesting because here we have just 67 OTUs of a total of 8,000 OTUs, but it's quite complex, the factors that are affecting arbuscular mycorrhiza. We can also test the effect of altitude, because we have an altitudinal gradient from around 600 meters above the sea level to 1,400. This in southern Chile, this is 40 degrees south, so this is quite high over there. This is the tree line, basically. There is no effect in all fungi, in saprotrophic and in ectomycorrhiza, but just in arbuscular mycorrhiza. It's a significant decrease of fungal richness of arbuscular mycorrhiza when you increase altitude. So to conclude this, the main result is that the abundance of ectomycorrhiza and saprotrophic fungi are negatively related. Okay, this could be explained by several things. First, there could be, of course, competition between saprotrophic and ectomycorrhiza for the different resources, but also saprotrophic fungi was highly abundant in conifer forests, and the litter of conifer forests is richer in nutrients, stays more time in the soil, while the litter in nothofagus forests is easily degraded, is poorer in nutrients. Then the fungal communities differ in the 2 mountain systems and in ectomycorrhiza and arbuscular mycorrhiza dominated forests, as we saw. They differ mainly in the community structures. I didn't show the results of, basic results of richness or abundance, which did not differ, but in community structure they did differ. And also different fungal guilds are differently affected by these different biotic and abiotic factors that we tested. By biotic factors, I mean the mycorrhizal dominance, and by abiotic factors, the mountain system or the edaphic conditions. Ectomycorrhiza and saprotrophic fungi seems to be more related to each other and more affected by the same processes than arbuscular mycorrhiza. And I finished to thanking the different funding agencies, and this is part of a bigger project called EarthShape that is in collaboration between Germany and Chile to see how this fungi degrades minerals in the soils, and by doing that produce new soil, basically. Thank you.
Thanks so much for that, Cesar. And I guess you're probably also the person that's travelled furthest. I don't know, that might not be the case throughout the US, but it's great to have you here. So we've got some time for questions now. If anyone wants to kick off, if not, I will. I'm jealous of your field sites, by the way. So Cesar, this negative relationship between ectomycorrhizas and saprotrophs is by kind of inference there's some kind of competition between these, or is it not that straightforward at all?
I will say that, at least for data of the region, we know that, for example, the Araucaria forest, the Fitzroya cupressoide forest, that is this huge conifer of 80 meters. The litter, the leaves are really good, had a lot of nitrogen, a lot of calcium compared to Nothofagus. So maybe it's more related with the litter, but of course in controlled studies, always this competition between saprotrophic and ectomycorrhiza has been noted, has been tested. So I think that they are not mutually exclusive hypotheses to explain this negative relationship.
Okay, excellent. So,
Yeah.
Tom,
It's just an observation. You seem to be seeing the same thing as Americans are seeing in the middle of this Indiana area, Richard Phillips's work, showing that litter quality is driving which from either AM or ectomycorrhizal fungi, driving the route of which carbon cycle. So you might want to look at some of the experiments that's going on in the USA.
Okay, thank you.
A lot of synergy.
Yeah, thank you.
so I'm intrigued by, you mentioned that the conifer litter is high quality in terms of nitrogen content and other minerals as well. I kind of would have, intuitively, I would have thought it's actually quite physically quite tough litter to degrade, because I guess there's quite a lot of waxes in the cuticle and so on. It's quite maybe quite thick litter structurally. I don't know if there's any sort of like aspect.
Yeah, for example, the Araucaria leaves, they stay like 30 years, so it's really strong.
Yeah.
But in some of these places we have 8 meters of precipitation a year, so that accelerates a lot. The climatic conditions are also really extreme. Yeah.
Yeah. That's even west than Liverpool.
Yeah.
Okay, so we've got a minute or two before we're due to start on the next one, and with courtesy to Jasper, I'd rather start on time. So actually, if anyone's got any particular points they would want to, thanks as well, that's fantastic. So any points that you might want to ask, because I realised that Wookey, you didn't have much time for questions, so Sigrid and Maria as well. If anyone's got any questions for them or one quick question, then please go ahead, or we can have a quiet pause for a moment while I load Jasper's presentation. Do chat amongst yourselves for a moment.
So Jasper, we'll just give people a minute. I think that we're running about a minute ahead of schedule. Just in case anyone wants to.
Thanks so much.
And then it's a bit embarrassing, I know what comes in. Exactly. We've got like 30 seconds. Come on in, folks. Take your seats. Okay, so you have the floor, Jasper. Welcome.
Thanks very much, Philip. It seems to have worked out to wait a bit, so thanks for coming, and good afternoon to you all. Thanks for bearing with us. I want to talk to you a bit about directing the development of ecosystems, of plant communities, and what soil inoculation could do in that, and particularly how we can use that in restoration. So as you know, many of the Earth's ecosystems have been degraded, but there's in fact also large efforts to restore degraded ecosystems. In fact, the EU has pledged to restore 15% of its degraded habitats by 2020. Now, there's many issues with trying to restore ecosystems. In many cases, it takes really a long time, and in fact, in perhaps even more cases, we're not reaching the targets that we would like to achieve. So if you sort of look at this red line, we're sort of straddling on, going somewhere, but not quite getting to the ecosystem, not quite getting to the species composition that we want to restore. Now, there's some common limitations. Many are the high levels of fertility that you find in sites. Others are that plant seeds or the organisms that you're interested in don't have the appropriate propagules anymore in the area. But one factor that's sort of ignored largely, and which may hold the key to successful restoration, might lie actually in the soil community. This is actually what we want to do. We want to go really quickly to the really good ecosystem. So, I don't have to tell you that the soil is really diverse. There's very many organisms living in there. There's a tremendous diversity. All the sequencing study is really crazy, the amount of OTUs that you are engulfed with, basically. But there's also many nice experiments that show what these soil organisms are doing to plants and how they're shifting the performance of plants. And I'm not going into the details, but it's sort of the upshot of many of these experiments is that if you change the microbial community or you change the whole soil community, that actually can change the balance of the competition of different plant species. And it seems to work out in the way that if you take a soil community that has developed over a long time, so it's from a late successional soil that hasn't been disturbed for a while, that this soil community is really beneficial to target plants that are usually also late successional plants. And they're in fact hampering the plants that are sort of ruderal, weedy species at the same time. So people have started wondering, well, what happens if we start playing with the soil community? If we start sort of, dialing at this switch, can we then actually shortcut maybe succession and restore our ecosystems much better? So this is what interested me and what has been the driving idea of my research project. But we wanted to test this really in the field, really go outside and sort of knock on an ecosystem and see what we could do with the soil biota. Now, one of the things we realized is that you do need to take account of the abiotics, so you do need to restore some of the abiotic conditions, I think, before you can do any proper restoration. So we did topsoil removal, which is quite an extreme intervention, but in Holland it's actually quite widely practiced. We have really high loads of phosphorus and also nitrogen in our soils, and if you leave that just as it is, you will never ever get a plant community that is oligotrophic, basically. And those are typically the plant species that we want to restore, so you need to do some abiotics interventions to get that going. But there's also definitely a role, I think, for the soil community. So this is what we started testing. What happens if we inoculate these areas with soil from well-developed ecosystems? We did this in the field. This is a former agricultural field that had been cultivated for, I think, 5 or 6 decades for wheat, mostly, also maize a lot. And then in 2006, it was given to a nature organization, and they said, oh, come on, we really want to do something special. We'll implement some of your ideas, and we'll start inoculating this area with soils. And they had the goal of restoring both infertile grassland communities and dry heathlands. So we went to that field, we removed topsoil, but we also, within these areas where the topsoil had been removed, we inoculated these soils from these 2 well-established communities, so really old dry heathlands and late successional grassland sites. And when I say soil inoculation, I really mean a very thin layer of soil. So we didn't transplant any entire turfs of soil from one side to the other. It was a really thin layer, usually about 1 millimeter, sometimes 5 millimeters in thickness, so really a very limited amount of soil basically per unit area, but we did it as a big scale. You could see it from the sky. We had different replicates, which you can see on the left, and within each of these replicates we had areas where we inoculated with soil from a heathland, with areas where we inoculated with soil from a grassland, and then we had these sort of set-aside control areas where we did nothing. The topsoil removal areas were 2 to 5 hectares in size, and the inoculations by themselves, they varied a bit in their size, but they were roughly half a hectare each. Now then after 6 years, we sort of waited and see what the community, how it would develop. And after 6 years, we went back and just sampled everything that we could think of. And I want to share with you what came out of that. And I think the first step is sort of just go there and see what it looks like. And that represents sort of the clearest image of what is going on. If you go to these topsoil removal areas where we did nothing else, where we just let the soil be and the plants be, you hardly see any vegetation cover after 6 years. It's basically still a desert. Whereas if you go 20 meters to the side where we did this inoculation, the situation looks quite different. We already have a quite nice head of vegetation developing there when we add a little bit of soil from a diheathland. Now, of course, we didn't only take pictures, we actually sampled the whole thing properly. This is an NMDS plot of the plant composition. You see the two donor communities in the top, they're well separated in the community composition, also the control areas at the bottom. But the interesting thing that we found is that, in fact, these inoculated areas, they're in between these two donor sites. But if you look closely, it's in fact each of these treated areas, the areas that we treated with the soil inoculum, they're already shifting in the direction of their respective donor community. So based on this, we thought it might be possible to direct the development of an ecosystem based on the inoculum that you use to kickstart your succession, basically. Now, we also look below ground, and they're also very similar to their respective target communities now. We also look below ground, so we investigated the microbes, and we also investigated the nematodes. So you see the microbes on the top and the nematodes at the bottom, and you basically see the same pattern. The control plots are on the left, the two donor sites are on the right, and they're well separated, and the inoculated areas, they are shifting in the direction of both of the donor communities. Now, then we wanted, and you're probably asking the same, is this not all due to the plant seeds that you're also inoculating with your soil? And we did a greenhouse experiment to test this. So we basically replicated the entire experiment we did in the field. We replicated it in a greenhouse. We took soils from the two donor communities, the heath community and the grassland. We inoculated into the field soil. And then we sowed a standardized mix of plant species on top of that. So we had ruderal species in there. We had target species from each of the two communities in there, some mid-succession species. And on each of the mesocosms, we sowed exactly the same plant species. So that was completely kept the same. And then we looked, I think it was 7 months we let it grow, 7 or 8 months. Later, we went back and sort of recorded the vegetation in these mesocosms to see what had happened. And to my surprise, we actually found exactly the same thing. So you see that the treatments here in this plot are again well separated. You have the control on the right, the grassland inoculum on the top, and then the heathland inoculum in the bottom left. And they're both shifting, if you compare the community composition to the donor sites, they're both shifting in the direction of their respective donor. So it's not only the seed community that you're introducing with the soil inoculum that determines where your ecosystem is going. It is also, in part, the soil community that is a driver of the successional changes and the direction this succession is taking. That plays a role in this. We also compared what all the species were doing and how the greenhouse and the field experiment compared to each other. We found very similar patterns. So the target species, both for the grasslands, which are on the left, and the heathland on the right, they tend to go up with soil inoculation, while the early succession species and the mid-succession plant species, they sort of more or less stay the same in both cases. So I think this has been a nice demonstration of what the potential of soil inoculations is in restoration and in directing the succession of plant communities. There are, of course, many questions that are still open. One question that I personally want to work at is how big is the influence that the soil community is, and does it depend on the community composition in the inoculum that you're using? So if you take, say you take soil from a late succession forest and then the edge of a forest and you know different ecosystems that will differ in the plant community in the soil community composition. To what extent can you then fine-tune the development of the community that you're trying to restore? And another thing that would be really cool is if we could use these things to combat different kinds of soil degradation, but that's still a long way off, but this is a personal dream of mine, so I hope in the future I will be able to work on that at some point. Then I want to thank all my co-authors, thank my funding, and of course you guys for listening, and I'm happy to take any questions. Thank you.
Thanks, Jasper.
Really neat study, Jasper, very tight also. And what I'm now just wondering, how does the the history of the site from today? Like you have a seed bank also, well you show that seeds are not important, but I can imagine that the history of the site could be important.
The history of the site is definitely important. This is also, of course, why we removed the topsoil, which is a bit of an extreme intervention. And I did not quite say that seeds are not important. I think they're key. I think it's both things at the same time. You need to introduce the proper seed stocks, basically, in order to restore the vegetation. Otherwise, it's simply not going to happen. But I do think our data shows that the soil community that goes with it has a complementary role, and that you need to do both in order to be successful. Yeah, so I came along, I think, in year 4 or 5 or so of the experiment, but there have been people measuring there before. Things go quite quickly. I think in 2 or 3 years. It depends a bit on the plants that you look at. For instance, the header plants are really slow-growing plants, so they also have very long germination time, so it takes a while for them to sort of kick in. So you need to give it a couple of years, but it's not that only in year 6 you would see something. Yeah.
Okay, so Luca first. Well, Ian, please.
So I spend a lot of time around soil mesofauna, and I'm just thinking that taking them all out in their pristine inoculant condition, have you had any extinctions? Because being kind of spread at 1 millimetre thin onto freshly dug soil is actually quite stressful for mesofauna. And while it seems to be heading in the direction of the donor sites at the moment, if you've had extinction of some of the more specialist and some of the more, let's say, sensitive mesofauna, you might find you don't hit that target because they're the sort of things that really define those ecosystems later on.
But I completely agree with what you're saying, and this is also what's evident in the data. The diversity that we find back in the inoculated plots is not quite the same as what we had in the donor soils. You will also see, if you compare the data sets of the different taxonomic groups, so the plants and the microbes and the nematodes, you see that the nematodes are in fact the groups that are worst responding, and they're known to be very sensitive to disturbance by soil handling and things like that. So that's completely in line with that. So I fully agree that you will disturb the community. I will fully agree that you will lose species. So this is, I think it's not a way to rescue everything. For instance, if you would say we have a very pristine area, it's just in a way we want to do some urban development or whatever, or we have the soil inoculation, we take the whole thing and chuck it somewhere over there. I absolutely believe that's not going to work. You will transplant some of the diversity for sure, but not all, and probably not the ones you really want. I do think this is a method that can sort of bring in key players that are needed in the beginning of the development, and then hopefully through dispersal or other means, things will start to come in that are more specialist.
Do you think you're going to need to do secondary inoculants for some of those things like nematodes and maybe some of the more sensitive fungi?
That will be interesting to look at. Yeah.
We've got 3 questions and probably time for well, we've heard from Luca. You can chat to Jasper later. Maybe a question at the back.
Last question.
Yeah, I think probably. Yeah, we might get
The
a
difference between the upper and the down picture is quite impressive, but did you have also controls without the topsoil removal? How did it look like?
I don't have the pictures here, but I do have the data. So we repeated part of the experiment without topsoil removal. Where we just inoculated with the grassland soil outside the topsoil removal areas. And if you look at these bars, so the light bar is with topsoil removal and the darker bars without, you see that the effect sizes are getting lower, so you're less efficient in transplanting the community, but it does still have a big and significant impact on community composition. Now, if you start digging into the data, what this actually means is you're adding species. So you're not kicking out many of the species that are there, but you're definitely introducing new things into those plots.
Could you maybe come back to Jasper after the session? So we should probably move on, I think. Yeah.
Thanks so much.
Thanks so much.
Good, so we got Michelle. Excellent. Okay, I think you should be all, here we go, I think this is yours. Yeah. Okay. So we want to go into. Okay, that's right. Thanks very much.
So I'm handing over to Michelle.
Yep.
Thank you.
Thank you. So good afternoon, everyone. Now we are moving from the mycofauna in soil, then move to the macrofauna. Then I'm Michelle. I'm from Hong Kong. I'm now studying in PhD, and I work on earthworms. I come from the Department of Geography and Resource Management from the Chinese University of Hong Kong. Before I start my presentation, I would like to, I think all of you know about him. He's the biggest man in ecology, Charles Darwin. And then in his last scientific book, it's mentioned that actually earthworms have played a more important part in history of the world. So how earthworms work so good in the ecosystem? Because they are the ecosystem engineer by increasing the aggregate size and also porosity of soils because they fit in soil. And then they also regulate the soil organic matter by changing the nature of SOM and also litter dynamics. They also help to stabilize the carbon and nitrogen in soil and in a result of to balance the soil greenhouse gas emission. So I'm sure you may all know about the mechanism or the processes of the soil greenhouse gas emission. The three major greenhouse gas, the carbon dioxide, nitric oxide, and also methane, can be produced from soil from different kinds of processes, such as soil respiration, electrification, or delignification, and also methanogenesis. In a meta-analysis study of lubus that he published in Nature, so he concluded that with the presence of earthworms, it can increase the CO2 emission by 33%, and also the NTO emission by 42% because of the aerobic respiration and also the enhancement of the electrification, and that's why NTO be produced. And then also in the earthworm gas, because it provides an aerobic condition for the bacteria, so it's also enhanced the production of N2O. As a result of the earthworm activities, they produce cast and also they produce burrows. And from this cast and also burrow wall, it also can emit N2O. But in the long term, it's concluded that earthworm may help to stabilize the carbon by stimulating soil aggregation. And from other studies, it's also found out that earthworm may help to affect the emission of methane emission. However, most of the studies they are conducted in the European or Western country, and also they work on the agricultural ecosystem. Because for the soil greenhouse gas emission, it is highly depend on the earthworm species and also soil type. So therefore, there's a huge research gap to the subtropical region. And when we look at the climate-induced environmental changes, actually warming temperature and also changes in precipitations has been recognized as the major impact to the tropical and also subtropical regions by the IPCC. Therefore, heat waves or the extreme precipitation event will be more frequent or intense in the future. Therefore, I would like to ask, so do the impact of climate change, just like warming temperature and also changes in rainfall, can affect the growth and activities of earthworms? And how the induced soil greenhouse gas be affected as well? And how the molecule of the soil earthworm-induced soil greenhouse gas under this impact or the stress on the climate change? So here comes to our simplified graphic to show my assumptions. So because of the climate change, it should have a warming temperature and also changes in precipitation. So it may have impact to the earthworm activities, which affect the CO2 and N2O emission. And because of the enhancement of the anaerobic condition by heavy rainfall, so the methane emission may also be enhanced. So the study site is in Hong Kong, far away from here. And this should be the image that you think about Hong Kong, high-rise building and so many people. But actually, Hong Kong, we have a very good environment and also have different kinds of land use and protected area, such as we have natural grassland, secondary forest, and also farmland, and also urban park. Although it should be in a small scale compared to UK. And more importantly, in Hong Kong, we do have many species of earthworms. In the first part of my PhD study, I conducted a community survey in the field, and then I already found that Hong Kong we have already more than 15 species of earthworms, so therefore it should be quite important. Therefore, I conducted a 30 days microcosm experiment to evaluate earthworm-induced soil greenhouse gas flushes under different temperature and soil water contact conditions. There's the treatment, there's a 3-level treatments with 3 replicate for each. There's with earthworms and also without earthworms. And I used an exotic one, which is the Isania fordita, because it's more easier to keep in the experiment. And also there's 2 temperature level control and also 3 soil water content control. And the soil was collected at Tai Mo Shan Natural Grassland, which is on the map. This is the highest point of Hong Kong. The soil type is clay loam. It's an acidic one with bulk density 0.93, so it's quite compacted. And gas sampling occurred in following days with 15 sample days in total. So here comes to my result. To understand the earthworm survival and growth rate, so I just hand sorted all the earthworm from the soil chambers after the study. So earthworms, they lost weight in all treatment, and the biomass decreased by 15 to 52%. And however, you can see most of the treatment they reproduce in the soil chamber, and then they also produce quite a lot of cocoons. So I conducted the 2-way ANOVA, but however, there's no significant effect on temperature and also the moisture treatment on the earthworm biomass lost. And here comes to the accumulative soil greenhouse gas emission for the 30 days experimental period. First of all, when we look at the CO2 emission, the cumulative one, we can see that actually temperature do a very significant impact on that, because most of the greatest emissions is emitted by the high temperature and also with the presence of earthworms. And when we look at the N2O emission, and it's also the same, temperature do a very significant job, and also you can see the 2 greatest emission is also produced by the presence of earthworms. However, when we look at the methane emission, actually the pattern is not that clear, and the highest emission actually is produced by the without worm treatment, and I will explain it later. So then we further deep down to see how the earthworm-induced soil greenhouse gas flushes. From this 3 graph, you can easily tell that for the CO2 and N2O emission, the presence of earthworms actually increase the gas emission very well. But however, for the methane emission, we don't see the case. Therefore, to have a statistical analysis, I conducted the three-way repeated measure for ANOVA, and it also tells you that temperature is a very significant factor on both of the three greenhouse gas processes, and also for the soil water content, it only affects the N2O and also methane emission. And when we look at the factor of earthworm, it only has significant effect on the emission of carbon dioxide and nitric oxide. However, all of this, the interaction all among these factors is not that significant. So therefore, earthworms actually increase the cumulative CO2 and N2O emission by 38% and also 58% in subtropical grassland soil. And it can explain it by the earthworm respiration and also the induced microbial respiration for the increased gas emission. And it's also in parallel with the result that from the previous study that the Ephedia induced 40% of the total CO2 production in microchromosomes experiment. However, when we look at the methane emission, in this study, it decreased by 59% over the 30 days experiment. However, the effect of earthworm is not that clear because you see the fluctuation of the cumulative emission is quite high, so maybe further study is needed. So warming temperature had a significant effect on the cumulative emissions, but they have no significant effect on the biomass lost. And changes in precipitations only have effect on N2O and methane only, because higher soil water content can enhance the nitrate availability of nitrate and also a higher delignification rate. So to conclude, we found that actually the warming temperature and changes in precipitation don't have a significant effect on earthworm activities. However, they do have direct impact on the gas emission. And then earthworm have a direct impact to the CO2 and N2O emission by themselves, but however, the effect on methane is not that clear. So for my next step, I would like to continue to analyze on this soil parameter and add these parameters to the assumption as well. And then I would also like to look at the earthworm burrows to quantify the earthworm activities, and also to repeat the experiment by using a native species, and let's see whether there's some impact on the invasive species to the ecosystem or actually the lot. So here's the reference for my study. So at last, I would like to thank all of you for your listening, and also my team for my research, because without them, I cannot conduct the tedious work. And thank you very much, and welcome for questions.
Thanks very much for that, Michelle. So, we've got time for a quick question. Please.
Thanks for a nice overview of your experiment. I come from the group where Ingrid Lubbers has started her work. And I know Astilia, the earthworm that you've been using as a composting worm.
Yeah.
But as I understood from your experiment, you've been putting it into soil. Do you think that the species of earthworm, in this case a composting worm, which normally only lives in organic matter layer, would have affected the outcome of your experiment?
Thank you for your questions. I totally agree with you, because for the earthworm that I use is a composting one. Therefore, for all the soil analysis, I only analyze the topsoil layer. That is the layer that they're more active there. And this is also the reason why I will repeat the experiment again with the native species, because I do agree with the assumption that maybe the compost worms do different behavior in the lateral soil, so that's why I would like to repeat it. But the limitation is, I don't know, do you know about the Asian worms that's always called the jumping worms? So they are very hard to keep in in a capture environment. So that's why at the first stage, I just use the non-native and also a more patient worms. And yeah, later on I will, now I just know more about how to keep them in the lab. So that's why I will repeat it later.
Okay, thanks very much, Michelle. We should leave it at that.
Okay, thank you. Thank you.
So next up we have Jean-Pascal, but he prefers to be called Pasky amongst friends, and I think we are all in the BES amongst friends. So there we go. Right, let's get you set up. Yeah. Okay, Pasky.
Okay, thank you. Well, thank you all for staying around even until the 11th hour. I hope you won't be disappointed. I know I won't. So, I'm Jean-Pascal. This is a presentation on some of the work I've been doing as part of my PhD. So let's start the story from the very beginning. Crop protection products, or CPPs, as I may refer to them for the rest of this presentation, are broadly defined as anything which is a herbicide, fungicide, or especially pesticide. So if a CPP persists in the environment after application to a crop, this can be potentially hazardous to both the ecosystem and human health. For this reason, regulatory bodies across the globe have strict regulations requiring a product to be intensely safety tested before it can be licensed for market. Most regulatory bodies across the globe, including the European Commission, have adopted the OECD regulation guidelines, the OECD being the Organisation of Economic Cooperation and Development, itself being an intergovernmental policy think tank. So these regulations involve tiered testing, which means a product must pass one test before it can move on to the next. And these tests look at hydrolysis, photolysis, phytolysis, and most importantly, microbial breakdown. The reason why microbial breakdown is so important is because it's the only process by which a product can be fully mineralized into CO2 within a realistic time span. So this is what the first test on the microbial breakdown tier looks like. It's called a closed bottle test, or to give it its exotic name, OECD 307. Not to be confused with OECD 301D, which is also called a closed bottle test. Common mistake. These tests simply involve adding soil into a bottle, applying a product, and monitoring the degradation over a set time course. These tests are carried out in the dark and at constant temperature and moisture in order to solely focus on microbial breakdown. However, before you can add the soil to the closed bottle test, the OECD guidelines require a set regime of processing in order to standardize all the tests. This involves air drying until it can pass easily through a 2mm sieve, and then moisture adjustment back up to what's known as its PF2 value, which is usually between 20–25% soil moisture. These soils, after they've been processed, can be stored for about 6 weeks on average. This is due to the weather-dependent nature of soil sampling. It's not always possible to get out to the field immediately before a test. And in addition to that, the OECD requires a product to be tested on at least 4 different soil types. So this can sometimes even necessitate importing soils. The tests themselves last between 1 and 6 months. So just a simple cursory look through the literature, you'll see that drying and sieving and re-wetting have already been shown to alter the structure of both the soil and the microbial community. So therefore, these tests may not necessarily be representative of the microbial breakdown observed in the field. Microbial breakdown itself is a very complex topic, but can be broadly brought down to 2 main factors: the bioavailability of the product to the microbial community. This is governed by the physiochemical characteristics of both the soil and the product itself. And the microbial community, as with all communities, is driven by nutrient availability and competition. So fungi are a key part of the soil microbial community. They are major decomposers and are thought to play a key role in CPP breakdown. However, at present, this is quite poorly understood. A few studies have found that whilst fungi have a much lower degradation rate than bacteria, addition of fungi to a bacterial community greatly increases the rate. So it's thought that perhaps that fungi are facilitating a rate-limiting step somewhere along the breakdown scheme. This ties in quite nicely to current phrase that fungi deactivate and detoxify CPPs rather than actually using them as a primary metabolite, as is the case in bacteria. So we're left with quite a few questions. What is the role of fungi in CPP breakdown? How does this OECD regulation processing affect the fungal community? And even though these bottle tests may not necessarily be representative of the field, can we use them to deduce fungal mechanisms of degradation? If you want to know the answers to questions 1 and 3 on the slide, I look forward to seeing you in Ghent next year when I'll be able to tell you a bit more. But for now, I will be able to tell you some of the answers to this question. What is the impact of this processing on the fungal community? Does this processing have a significant effect on the community? Does the diversity increase, decrease? And more specifically, what are the taxa changes? So, to answer this question, I went out to 3 field sites over the course of 3 seasons last year. My field sites were Leeds University Farm. I can see we've got a few Leeds University Farm fans in the audience right now. As the name would suggest, it's an agricultural farm owned by Leeds University where many experiments take place. 18 acres is a pasture land next to Gellets Hill site owned by Syngenta and is the workhorse soil of the majority of their degradation studies. And Warngate Stray is another pasture land, a staggering 10-second walk outside the biology department here back in York. At each field site, I had 2 sampling areas, and from each sampling area, I took 3 cores. I subsampled each core for a effective T0 DNA extraction, and the remainder of the core went on to be processed. So, as I mentioned before, this involves air drying until it can comfortably pass through a 2mm aperture sieve, and then it was moisture readjusted back up to its PF2 value. These were then bagged up and stored for 6 weeks at 20 degrees Celsius in the dark. After that storage time, they were put into the degradation rig, which looks something like this. Air is pumped through water in order to moisturize it, then the moisturized air is pumped over the sample to keep it aerated throughout the entirety of the experiment. These were left in for 1 month at 20 degrees C in the dark as well, and no protection product was applied to this rig. This was purely to look at the physical parameters. I extracted a DNA using a power soil kit from MoBio, and then used PCR to amplify up the ITS region. The ITS region, if you're unfamiliar, is a classic fungal barcoding area. So I sequenced up the things that I amplified and went on to my bioinformatics pipeline. I clustered OTUs using usearch, and then once I had my OTUs, I calculated out the Shannon's index, the Bray-Curtis dissimilarities, and to obtain taxonomic data, I used the Unite fungal ITS database. Here we have some nice error messages I ran into. So here's some of the results which I've got. This is an ordination plot of the Bray-Curtis dissimilarities. If you're unfamiliar with the Bray-Curtis dissimilarity, it's simply a metric of how different 2 communities are. So on this plot, each dot is a sample, and so the further apart 2 dots are, the more dissimilar their communities are. Here they are coloured by field site. So in red we have the Leeds University Farm, and in blue and yellow we have Warngate Stray and 80 acres respectively. We can see nice clear clustering according to soil type on the diagram here. This is unsurprising, as it's been shown before that different soil types will have different microbial communities, and a Permanova showed that this was having a significant effect. If I recolour the dots now to be the treatment with the processed soils coloured red and the unprocessed soils coloured blue, and simply rotate it around, we can start to see some clustering again. It feels prudent to point out these ellipses do not have any confidence intervals attached. They're simply just visual aids for the purposes of this presentation. A Permanova revealed, yes, treatment is having an effect here. And I think the key thing to take home from this ordination here is that the processed samples are clustered much more tightly together than the unprocessed samples, suggesting perhaps that processing is biasing communities towards a certain kind of assemblage. A cursory look at the alpha diversity reveals that there is no significant increase or decrease here, so any changes we're seeing in the community is down to a taxa shift. So, at the table at the top here is the top 10 most abundant taxa in the unprocessed samples, and the table below it is the top 10 most abundant taxa in the processed samples. Starred taxa appear in both tables, and these arrows here indicate whether or not the taxa have gone up or down in abundance after being processed. These are color-coded by their function, so taxa color-coded in red are taxa which have been linked to saprotrophic activity. Those colored in blue have been linked to chitinase activity and chitin saprotrophic activity, and those colored in green don't fit into either category. Really, the key thing to take home from this is that we're seeing a dominant shift towards taxa which are saprotrophic, and especially those which are able to break down chitin. This is of special interest because it would suggest that they're able to actually break down hyphae that are in the soil, which would suggest to me that nutrients are becoming a limiting resource within these closed bottles, for biasing the community towards those taxa which are essentially fungal cannibals. So, yes, we can see that processing has an effect on the fungal community. The alpha diversity remains unchanged, and the taxa shift we are seeing towards saprobes and possibly hyphal saprotrophs. In order to pull out some more meaningful conclusions from this, I'm going to separate the samples out by location and season and examine them independently to see whether or not this effect of processing on the community is predictable. Does this happen the same way each time, biasing towards the same assemblage independent of location and season? I'd like to thank the BBSRC for funding this work, as well as Syngenta, the Helgeson Lab Group for their continued support, the D0J1 corridor team back at York for keeping me sane, pictured here having a grand old time last Friday, and Arena and Robin and everyone at Syngenta Product Safety for their continued support as well. My contact details are here. Please do follow our lab on Instagram if you like pretty pictures. Thank you for listening, and I'd welcome any questions.
Thanks very much for that, Pasky.
No worries.
So it just goes to prove that Helgeson Lab can have a good time without any alcohol as well.
Yeah, not pictured there.
We've got time for a couple of questions, I think. So please go ahead.
Have you thought about one of the problems with the Bray Curtis index is that it can reflect species turnover and nestedness in your communities. So have you thought about, if you look at the work of people like Andreas Paselga, where they've managed to partition those sort of indices into a nestedness component and a turnover component, whether that help you pick apart why?
Yeah, something which I've thought about. I haven't found exactly what I'm going to do with this, but a key thing to bear in mind here is that these samples are nested. So the T0 samples are all subsamples of the complete course. So anything that I need to do from this point onwards, I need to bear in mind everything is nested.
Time for one more quick one. Okay, I'm just going to jump in then. So basically this pre-treatment, would it be fair to say that you're effectively selecting against the bacteria relative to the fungi overall? Desiccation tolerance, I'm thinking more specifically.
It's possible. I haven't actually measured any of the bacterial stuff here. In my future experiments, I will be looking at both fungal and bacterial communities to try and track what's going on there. But definitely, I think with desiccation resistance, it possibly would tend towards more fungi, which would highlight, again, the importance of fungi in these closed bottle tests.
I guess Syrian's another issue then.
Yeah.
Thanks very much then, Pasky. So
Thank you.
on to the last but by no means least presentation. It's Ashley for this afternoon. And
Let's see if we can get you set up. That's the one, isn't it? Perfect. So over to you, Ashley.
Thank you. Yeah, so thanks for staying to the bitter end. I'm going to apologise up front. There's no fungus in this talk, but hopefully for 10 minutes I'll be able to convert you to some seed ecology. So I'm a PhD student at the University of Dundee and the James Hutton Institute, which are in Scotland, in case you're unaware. And my project's funded by Chequer Centre for Environmental Change and Human Resilience. Got my Twitter details up here, and also if you follow Chequer on Twitter, it's a pretty good thread. So, I'm going to be talking about how arable weeds can influence the soil structure. 2 seconds. How arable weeds can affect soil structure, and how we might have potential beneficial morphological properties that we can use to bind soil particles together and prevent runoff erosion in fields. So first of all, arable weeds are really important, much to a farmer's disbelief. They provide important ecosystem services within simplified agroecosystems, so things like nutrient recycling, moving pests away from crops, but also for crop pollination. I'm sure I don't need to say this, we've heard it all week, but 35% of crops require crop pollination, and researchers last year at the University of Reading value this to be worth £690 million just in the UK. However, accelerated soil erosion can degrade the physical and biogeochemical functioning of soil, and so weeds then might become a vital buffer against short-term disturbance events. They are influenced by cultivation practices, and we've seen over the last 50 years a decline in biodiversity around the country because of intensified agricultural management, which in turn makes arable weeds really effective indicators of farmland biodiversity. My main research question is looking at how seed banks can influence the physical and biogeochemical behaviour of the soil. So some weeds have important morphological properties which might be able to benefit soil structure and water retention. And so I want to investigate these seed-soil interactions as a natural solution to preventing runoff erosion in fields. After Storm Desmond, we can see in this picture here, so Storm Desmond was the end of last year. These are small tillage experimental plots that we have on site at the Institute, and you can see how the tram lines become a superhighway for the transportation of biota, for seeds, but also for pathogens, and also any chemicals that the farmers sprayed on the field as well. And then when we upscale this to a farm here in Montrose, which is north of Dundee, it's a cause for concern. Dispersibility of a seed depends on their morphology, so this causes selective erosion of the seeds as well, based on their size, their shape, whether they have hairs, whether they have wings, and then also this trait here, which is called mixospermy, and this is the trait that we're focusing on in my project. So what is mixospermy? It's a common trait among arid and disturbance-loving species, which makes them prevalent in agroecosystems. The trait is, in the general sense, the secretion of mucilage. So I'm just going to run through the process of how the secretion is released in the seed. So first of all, we start with a mature seed. It becomes hydrated. Then a volcano-shaped cytoplasmic column then forms in the center. The mucilage then ruptures the primary cell wall. The columella, which is the secondary cell wall, then gradually displaces the cytoplasmic column. And then lastly, we end up with a two-layered structure where the inner layer is adherent to the seed and the outer layer is non-adherent to the seed. And when the mucilage dries, we think that this has the potential to be able to glue the seed to the seed particles. One vital part that I should make you aware of is that this mechanism can occur even if the seed isn't viable anymore. So when we think of this from a commercial point of view, the farmer's not going to want us putting a bunch of viable seeds into the soil, but it can do this process even though they're not viable anymore. So he'll be happy to hear that. In terms of the composition, we don't know a lot. So I'm going to use Capsella here as an example. It has a classic halo shape, this nice pink bubble around the seed. And in the inner layer, it's comprised of pectin, so mostly RG1, and it's non-adherent to the seed, so it's soluble in water, which means it's going to have the potential to float off into the soil matrix and alter the structure. Whereas the inner layer is comprised of cellulose fibers that are they're tightly packed in this pectin matrix. So it's just these rays here coming out of the seed coat. And this layer is adherent to the seed, so it's only going to be able to affect the soil that is physically surrounding the seed itself. The chemical composition of this mucilage is largely uncharacterized for many species. At the moment, Arabidopsis is the only one that has been analyzed in full. And for this project, we're not going to be doing any chemical analysis. The only way we're characterizing the mucilage is by microscope images like this, and then also a digestion method that I'll explain in a moment. So what do we know? Quantitative studies are rare, and they're usually limited to a single species. But what they have suggested is that there's ecological significance for germination, for dispersal, and dormancy. And that due to the adhesive nature of the mucilage, we think that this can cause physical alterations to the soil in terms of stabilizing structure and enhancing water retention. But what we don't know is the capability of specific species, and then also as a community together, whether they can then prevent their removal, but then also the removal of soil in a field. And also whether there's a threshold beyond which soil behavior and the biogeochemistry is altered. So not all seeds have this trait. In my project, I'm looking at 7 common agricultural weeds that can be found all around the UK. And what I didn't expect to find was how the mucilage structure of each of these species differed. So I'm just going to go through these one by one, some nice pretty pictures. This is Arabidopsis. It's the smallest seed that I have out of the 7. And you can see, very similar to Capsella, it has this nice halo shape. And you can just sort of see the cellulose rays coming out of the testa there as well. At the moment, we're developing a methodology for imaging the cellulose, so unfortunately I only have the pectin pictures to show you today. So in this picture, it's stained with ruthenium red, which binds to the pectin, so that's what you're looking at. Capsella, I showed you that a couple of slides ago, so it's very similar to Arabidopsis, but it's a much bigger seed. Plantago is the largest seed that I'm looking at, and at the moment, Plantago is really only used in arid and dryland restoration projects. So they have the opposite problem for us. They're too dry, so they want to put Plantago into the soil to regenerate the water content in the soil. We're coming at it from the opposite end of the spectrum. We're too wet. So yeah, we're doing it to trap the water in certain places in the field rather than it flowing off into the water systems. Senecio is the only rod-shaped seed that I have. And it's conical as well, so it also has hairs. But the structure of this mucilage is very fibrous. So if I had a cellulose picture here to show you, you would see it kind of looks like a spiky caterpillar type shape. Artica has very little cellulose at all. It's mostly pectin, and it's not uniform in shape like we've seen with the others. And then Veronica, it's there, but it's barely there, and it almost has no cellulose at all. And then lastly, Viola, a nice substantial halo shape, and again, lacking in cellulose fibers. So the extraction method that I briefly touched on is what we're using to characterize the differences between these species. So we're quantifying the inner layer and the outer layer. So to quantify the outer layer, we're doing a water dispersion. So it's soluble in water. This is what happens afterwards. You can see that we've reduced the halo size quite substantially. And then after an enzyme digestion, you can see that we've got rid of everything. So these are the treatments that we're going to be using in my experiments. So when I applied this to all 7 of them, you can see how the production varied across the 7. And surprisingly, Senecio was the most productive. So if we remember what they looked like, it was a thin, long, elongated seed. So obviously this is telling us something about seed surface area rather than seed weight. However, it wasn't statistically significant compared with Capsella, but we'll still have a nice comparison here because of the structural differences between them. And then Arabidopsis and Artica weren't different from each other, but again, we have the structural differences, and then not surprisingly, Viola was the lowest producer of mucilage. So now we have a set of species that have different productive mucilages and then also different structures of mucilages. But what will this tell us about their interaction with the soil? So the way to answer this question is to start small, thinking at the aggregate scale. to look at these relationships between seed surface area and how sticky the mucilage is. So we'll put a seed in the soil once it becomes wet. Hopefully it will then bind to soil particles. And here we can think about the capability of the mucilage to draw in these soil particles and adhere them to the seed, creating an aggregate essentially. And then we might be able to identify a zone of influence, so how far out into the matrix can we see the mucilage, and then also whether it can then attract a secondary layer of soil particles around the seed. And then we can also look at the interaction that's occurring in these pore spaces in terms of the biogeochemistry as well. We can also look at the differences in particle sizes that are attracted to the seed. So will the soil particles be larger than the seed itself, or will we see a mixture of sizes? Will they be smaller, uniform, that kind of thing? And then taking out all of the information that we learned from that, we can then apply it to a small-scale runoff erosion experiment when we have a rainfall tabletop, piece of equipment, I think is the word I'm looking for. And we can use different slopes and also change the soil texture. We can apply communities of mixosperma species and non-mixosperma species to look at the differences in the soil that's been effectively washed off of the surface, and then also the abundance of seeds that have been transported as well. And the hope that we can alienate specific communities that will be able to reduce the risk of erosion. So we know about the abundance and distribution of these species because I have some data from my first year experiment which looked at tillage disturbance and the distribution of the seed bank in the soil. We now know the differences in production of this mucilage and also the differences in structure between these species as well. We will then have a clear idea and also visually be able to image the mechanism that's occurring at the aggregate scale, and we'll be able to see this zone of influence around the seed. And then we can use this to predict soil export fluxes and start to think about landscape biosecurity for soil and also implications for the wider ecosystem in terms of biodiversity and all of the knock-ons that will then happen to the microbe community and also straight up to crop yield. So to conclude, weeds provide important ecosystem services within monocultures. We're exploring this underlying mechanism and how it interacts with the soil. Can we then manipulate this mechanism to retain the soil structure under erosive conditions? And at the very least, we'll have a better understanding of the biophysical linkages that are occurring in the seed banks, relating that to land management, which ultimately drives policy of food security and sustainable agriculture. So I'd like to thank my wonderful supervisors, Tracy for helping me with the imaging, and the lab staff at the James Hutton Institute, and I'm happy to answer any questions.
Thanks very much for that, Ashley. Ashley obviously gets the prize in this session for the best Christmas outfit.
Thank you.
Unless there's anyone that I've overlooked. Okay, we've got time for a couple of questions. Okay, since we haven't actually mentioned fungi,
yes.
do you think any of this mucilage might actually be important in attracting mycorrhizal?
As far as I'm aware, they don't eat the pectin. You can all shout out if that's wrong. Bacteria will eat it though. So post-hoc proposal, once we've nailed this mechanism, I'll then be able to introduce the microbes into the system to see if we're actually altering the food webs within the soil. Because obviously we don't want to be altering it in a way that's going to be detrimental to the crop, you know, if we're promoting the bad guys over the good guys. So yeah, we'll have to test retention time and things like that to see, you know, how often would we need to prime the soil with the mucilage to make it effective for soil structure.
Thank you very much. Question over here?
It's a bit speculative and hand-waving.
I like those.
If this is a useful trait for sticking soil together, erosion in agricultural fields is such a problem, is it something that actually crop breeders should be breeding into crop lines?
Yeah, essentially. Yeah, exactly. I think I would, if we ever get to a commercial point, I will, you know, call it something that's not weeds, unless we've changed our view on the word weed by then. But yeah, I think commercially, we'd also need to test whether you actually need the seed itself Because if we can harvest the pectin and the cellulose combination, whatever it ends up being, we can either sell it as a spray that's sprayed into the tram lines, or we can even synthetically produce it as well. So it depends on whether you then need the seed for the, the morphology, the shape of the seed to then create the pore structure, or whether you can actually get away with just spraying it. Because if you can just spray it, then you're going to avoid the confrontation of, I'm not putting weed seeds in my soil kind of approach. But yeah, we're miles away from being able to do that, because there's nothing being done on this at all. Plantago gets used for the opposite direction. Arabidopsis is used from, genetics side of things. Obviously, it's mapped. They know how the mucilage is being switched on and turned off. And a previous student at the Hutton just used Capsella looking at physical properties. So, yeah, we're all in the dark with it. So it's fun, though.
Okay, thanks so much.
Thank you.
Ashley, and thanks to all of the speakers this afternoon for a superb range of, fantastically presented clear presentations, as well as the geographical and subject coverage, which was fantastic. So thanks.